I have the honor to introduce our last speaker for the afternoon session, Dr. Kailas Thakker. She's the president of Topical Consulting Firm. Kailas founded Analytical Solutions in 1994, which was later rebranded to Targus Pharma. Targus Pharma is the world-known service provider for topical product development. In 2021, Kailas launched her consulting firm named as Topical Consulting. She also serves as a chair for USP's expert panel for topical and transdermal dosage forms. Kailas, the podium is yours. Thank you.
I'm going to talk today about the considerations in development, validation, and transfer of in vitro release methods. You've heard a lot about it in last several presentations. My focus is going to be on the challenges that we have experienced over the last 25 years of my working with IVRT methods, and how do we overcome, if at all, these challenges. So all the experimental data that I will be presenting today is generated either at Analytical Solutions or at Targus Pharma, where I was a founder and co-founder of these companies, and IVRT was my responsibility. Next slide, please. So we have heard a lot about development of IVRT methods. I just have this here as a refresher. I'm not going to go into details, but suffice it to say that selection of the receiving medium and filter membrane, appropriate selection of apparatus, and the dosing technique and duration of the test will all allow you to develop a good validatable IVRT method. I want to show you an example. In my next slide, here, this is one example where we had one same generic product, and we were able to develop 5 different IVRT methods for using same principles of the selection of the different parameters. All 5 methods were validated according to acyclovir guidance, and we were also able to provide pivotal comparison. As you can see, the receiving medium range from hydrochloric acid to buffer to combination of hydrochloric acid and IPA. The solubility of the API was not an issue. We were able to use all different membranes, and they were all inert. The cell volume differed only in one case where it was a 12 ml cell, which is an older model of the vertical diffusion cells, and the same model also had a higher surface area of exposure. And because of that, we were able to use apply the dose using a syringe, using a spatula, whereas in all other cases, because of the geometry of the cell, we had to use syringe application, and the amount dose will range from 400 milligrams to about 1.5 gram, and that was mainly to manage the dose depletion. This kind of relates to the the arrangement of matter within the formulation. We were able to really come up with good methods. And go to the next slide, please. Yeah. So in my experience, what are the challenges that we get in developing a reliable IVRT method? The most common and frequent occurrence is that when we are using hydrophobic petroleum-based ointments, they present a substantial challenge. The IVRT method, generally, we are not able to develop a appropriate IVRT method because the release typically is very, very low. There is no increment of release with the time, so it's what I call a flat release. So we can get a linear release, but it's completely horizontal to the x-axis with just small amount diffused at the beginning of the test. The other example is when the drug substance is in suspension form, and it gives an additional challenge depending on these suspended and soluble ratios. Sometimes we have difficulty in achieving steady state. What can happen is that the soluble part of the dosage form quickly diffuses into receiving medium, then the dissolution of the suspended particle in the formulation matrix is very slow for some reason, and that does so therefore there is no any increase in the release, and you really don't have a steady state. So that can pose a great challenge. Challenges associated with the dosing technique, especially when you're using a syringe, how much here is applied to the dosage form can have a significant effect on your race and can cause variability. And this particular aspect I'll discuss a little bit more as we go along. Next slide, please. So how do we overcome these approaches how do we overcome these difficulties that we face in developing IVRT methods? We at Turgis had used many approaches. Some of these I've listed here. We use extended release time. For example, we take it out to 24, even 48 hours if necessary. And if it's just a slow release, that's not an issue as long as we get a linear steady state kinetics. Sometimes we use surfactant-based receiving fluid. This was a question that came up at the earlier panel discussion. And my caution is that the surfactants are okay to use if necessary, and they might be useful also when you can't use hydroalcoholic solutions. But one has to be careful in the amount of surfactant used, so that their surfactants typically can form bubbles, and because the receiving chamber are small in vertical diffusion cells, that the bubbles can form and lodge under the membrane surface, then there's no diffusion, and you pretty much beat the purpose of using that in the first place. We even thought of, in case of hydrophobic ointments, we even thought of breaking up the matrix, hydrophobic matrix, using high ionic strength fluid, receiving mediums, using buffer acids, bases, etc. We even tried using very strong solvents, stronger than your typical hydroalcoholic mixtures, such as DMSO, DMF, and in some cases we were not successful. Using and penetration enhancers such as isopropyl myristate to drive the gradient, and soaking the membrane in isopropyl myristate would sometimes help the gradient. In some cases, emulsion dosage form, we use membranes with slightly greater pore sizes. And last but not the least, we sometimes use increased temperature in the receiving chambers. Next slide. Next slide. So next, go back, please. Still one more. So here is one. I want to show you an example of a hydrophobic ointment, hydrophilic API that was formulated in hydrophobic ointment matrix. We had a particular difficulty in coming up with an IVRT method for this particular dosage form. The API was soluble in hydrochloric acid, sodium hydroxide buffers, etc., and solubility of meeting sink condition would not have been an issue at all. But we did not get any luck with the obtain a good linear release rate that had a slightly good sharp slope. So as you can see here, we have used combination of alcohol, acetonitrile, sodium hydroxide, DMSO with and without twine and sodium lauryl sulfate. We used sodium chloride solution. We used membrane soaked in a solvent, a strong solvent, and we used all different types of membranes. We went up to 37 degrees in one case and took the duration of the test over 24 hours. And this is just a 1/3 or 1/5 of the amount of effort that went into developing the IVRT method. So just give you an example how challenging it can be. Your typical IVRT development protocol should not need more than maybe 3 or 4 initial screening of receiving media if you know your solid and you know your composition. Next slide. So the other challenges that come across in developing reliable IVRT method is when you have difficulty in achieving specificity and sensitivity at high concentration API, not possible due to solubility limitations. So there, not much you can do, but you can have a slightly different concentration of API, whatever is allowable, and talk to agency, have a control correspondence, and it should be possible to come up with some kind of agreeable approach. Sometimes you develop an IVRT method, you have a nice linear rate, you have a wonderful precise method, and you come along and see that the release rate is pretty much not dependent on any of the parameters or physical parameters or any of the change in composition. That can pose a problem, because then you may not be able to validate for selectivity, and there you really have to either go back to the drawing board and develop different IVRT method conditions or evaluate a different dosage form. Next. So before yes. So when we try to overcome these challenges, it's important to understand what are the parameters that affect the rate of release. Now, we have all throughout today and yesterday, and we have gone through what kind of factors that affect the release of an active ingredient from a semi-solid dosage form. I'm not going to go into detail. I just have this here for you to see, but there are factors that are to the API characteristics, dosage form characteristics, and physical characteristics that affect the release of API from the dosage form. Don't forget the presence of some of the excipients that can significantly cause change in release rates, such as emollients, penetration enhancers, viscosity builders, etc. And you have your manufacturing process, depending on how much the order of addition, hold time, the way in which API is incorporated, as such process-related parameters, your manufacturing equipment, manufacturing site, all these have an effect on the release rate. Next slide, please. So here I'm going to quickly show you some examples of what we've come across during development of some of the products, where this here is an example where the grade of the API had a release or effect on the release rate, and it may be due to the different polymorph, maybe different particle size, but necessary for the formulator and the scientists to know that the release rate can be significantly different should you change the API. Next one. So another example is there where the sodium hydroxide concentration changed from 5 to 7%, and that would have a different pH of the dosage form, but there was no effect on the slope of the line slope of release rate of the API, because these lines were all parallel, so it was completely insensitive. Next slide. This is an interesting application where we were developing a lotion dosage form, and we had a beautiful IVRT method. It was linear and met all the other criteria. But then when we came to see the sensitivity using 150 and 150% of the product, we lost the sensitivity. As you can see, the 150% was slowly had a lower release than the 100%. Going on to the next slide, when we changed the forming agent on concentration in the dosage form, we were able to come up with, we were able to use this IVRT method, and it was sensitive to as needed. Next slide, please. So sometimes in this case, the composition, gelling agent composition changed significantly in the product, and we saw that the gelling agent had absolutely no effect on the rate of release. Next slide. Similarly, in this case, the API was partially suspended, so we wanted to see what type of effect the release rate has on the particle size of the API, and we were surprised to see somewhat that the milled and non-milled particles API. The dosage form had absolutely no different in the release rate. That was next slide, please. the same product, when was subjected to higher temperature, had a change in viscosity, and that change in viscosity was picked up by the release rate, and we were able to see a decrease in the release rate. So all these examples show that, these were all development activities, and these are not generics, but regardless, these experiments, all the examples all show that one needs to understand what kind of parameters affect the release of your API from the dosage form, and that can help you in the long run for many other future activities. Now, next slide, please. So, let's going on to the challenges in validation of the IVRT method. Validation, these are the parameter attributes that need to be validated for a good IVRT method to be used in a BAB comparison or any other super-related activity. Next slide. So first, going on to the first aspect, first parameter, first attribute is precision. The percent RSD of slopes for 6 cells, is acceptable is 2 to 15% of our RSD. But if 15% is allowed by the acyclovir cream guidance, but if you have a trained analyst and good, properly developed IVRT method, you can easily get between 2 to 10% or even less sometimes. But the precision is directly related to how the dosage form is applied onto the donor chamber. And consistency is a key here. There are 3 different types of operations that are used for applying the dosage form. That you just saw Christina show you the 2 techniques, one with spreading with spatula, the other is application with a syringe, and the third one is the use of transfer tubes. So improper spreading technique can introduce bubbles within the formulation if you're not careful. Sometimes the syringes can induce variable shear from cell to cell. You can squeeze more or less, and that can give you a difference in release and loss of precision. Application of transfer tubes is relatively new in my experience, and it still stores a lot of promise. Now, inconsistencies in all these techniques definitely cause variability and loss of precision. So the training is very important here. So if it's a trained scientist can be consistent from one cell to the other and pay attention to it, then we can have a good, precise IVRT method, and that easy to validate. One point I want to make here is that even when you're spreading, even with a trained scientist, when you're spreading hydrophobic matrices onto the donor chamber on a membrane, remember, membrane is generally mounted before adding the dosage form. At least that's the way we do it. And so the membrane is slightly wet with the receiving medium. The hydrophobic matrices are very difficult to be applied on a membrane, and it is very important to establish a good contact between the hydrophobic matrix and the receiving medium. This is one of the leading cause of loss of flow and loss of precision in case of some of these dosage forms. So one has to be extremely careful of applying these kind of matrices. If there is an inhomogeneity in the dosage form, then there will be a loss of precision also. For intermediate precision, as you can just envision, the differences in the techniques between operators will lead to loss of precision, a loss of in intermediate precision, and this has happened to us many times when someone has a slightly stronger hand squeezing out a tube very strongly versus mildly. And in fact, in one case, we had a protocol of actuating it so many times only with so much pressure. And that's how we were able to control the shear applied to the dosage form. No matter what you do, you always apply some shear when you're using the syringes. And differences in environmental conditions also will lead to loss of precision, where the release rates are very sensitive to temperature and environment, even if the donor chamber is occluded. Next slide, please. So other challenges in validating IVRT methods are when you have a properly developed IVRT method, and you have a good handle on sensitivity, specificity, and selectivity, validation is generally not an issue, because by then you already are able to validate it, except in the case of, let's say, when you have developed your method using a laboratory batch of development batch, and everything is fine, you're 100%, you're 50%, you're 150%, give you good sensitivity, linearity, whatever. When you come to do validation, you have to use GMP batches that are manufactured at a larger scale. I've seen examples where the different batches that are manufactured at different scale can have differences in release rate. So then your 100% batch is at a higher scale manufacturing scale compared to the 50 and 150% batch, and you may have a loss of sensitivity there. So I think it is recommended that for this particular validation of this particular attribute, that one uses all 3 strength batches manufactured at the same scale and by the same process. And I emphasize the scale and the process both, because that both can have an effect on the release of the API. In validating robustness of the IVRT method, most important parameters are receiving medium composition and membrane. So receiving medium, when using, we obviously use hydroalcoholic solvents all the time. So composition and preparation have to be very consistent, and in some of the API have a very high solubility in organic solvents, and the release rate is very sensitive to the composition of the or the percentage of the organic component of the receiving medium. So it is important to make these receiving mediums accurately and consistently, and volumetric measurements are desirable. In case of the filter membranes, it is important to know that different suppliers of the same membrane can have a completely different composition of the membrane. This has happened to us more than once, that you develop your method, or even validating your method using one lot of supplier, and you run out of that. You go into a different supplier and you see a completely different release rate or no release rate. So, but then you go back to the same original supplier, you get back to your results back. So I think it's important to know these robustness aspects of your IVRT method and sensitivity of the release rate to different supplier membranes, and keep in mind, and should you want to use the IVRT method for any future applications, one needs to make sure that you use a supplier that's going to be available in future, and that you have a good supply of that, at least for your validation and your pivotal comparison. Next, please. So transfer of IVRT method is not done, to my experience, that much yet. And I think I cannot emphasize enough the importance of keeping all these small details of an IVRT method in order to have a successful transfer. What are the considerations of successful transfer IVRT method? First, types of apparatus. So volume of a receiving chamber. We need to have an exact volume of the receiving chamber, and we need to know that, not the nominal volume. Surface area of exposure also needs to be known, and has to be the same, actually, and that also needs to be determined ahead of time. The geometry of the donor and receiving chamber is important. Even if you have the same volume, but the geometry is different, your hydrodynamics are going to be different. And so therefore, you will see differences in the release rate. And it's important to pay attention to that before going on to transfer method. Position of the sampling arm is important, because the sampling should be done right underneath the dosage form, and not in any part of the sample arm, because there may not be proper mixing in the sample arm, and that can cause to failure. What kind of stirring mechanism is used at the facility? That's important to keep in mind, that you use the same type of stirring mechanism. Dosing technique, I cannot emphasize enough. We've just gone through that and more than once shown how the dosing technique is so important in maintaining precision and accuracy of IVRT methods. The type of syringe, the type of how even it have the same type. How do you apply the dosage from inside on the donor chamber? Do you put a blob of it? Do you go in a concentric form, go inside out, outside in? All these small factors will lead to some failures if not paid attention to. How is the dosing chamber occluded? It's important. Next slide. So here I'm showing you we did some comparison of hydrocortisone cream using different cell volumes. Everything else was the same, where all other parameters were controlled. And we can see that with a different volume of cell going from 8 ml to 15 ml, you have different rates of release. It's okay to have that, but one needs to know that this is going to happen. If you have a different cell volume, you're going to have a different release rate. So don't be surprised. Next. So how do we successfully transfer an IVRT method? These are all my opinions, and I just think that this is probably one way to successfully transfer IVRT method. First and foremost, I cannot emphasize enough the importance of training. We have seen both at Thakker and Analytical Solutions that training scientists for proper IVRT execution is absolutely important. So the transferring facility has the most experience with the development and validation of IVRT method. If it's a CRO, they have experience in it anyway. So that facility needs to train the personnel at the facility where the methods be transferred using either a PVT such as hydrocortisone cream or the drug product, and with a very robust training protocol. Then once the training is completed and successfully completed, then the transfer can occur between 2 6-cell IVRT runs can be conducted using the same lot of product, conducted both facilities within the same week. We don't want to have a situation where something is, any environmental, any dependency of the release rate on environmental factors can cause a failure. Although if you're in a different part of the world, that can happen anyway, but at least we can try. Once the transfer is completed, the data sets are compared using an appropriate statistical method. I just put here Wilcoxon Sum-Mann-Whitney rank test, but any other appropriate statistical method can be used to compare the data sets, and one method is successfully transferred when the two data sets are equivalent. Next slide. Thank you everyone for listening to me, and I want to take this opportunity to thank the organizer to allow me to speak to you all, and also want to thank the 2025 scientists of IVRT team at Thakker and Analytical Solutions who have had invaluable contribution to what I have just presented to you. Thank you.